Hello everyone, welcome to Get My Class. Today we're going to solve this question which says, which of the following enzyme is molecular scissor? Option A, BAMH1, option B, RNA polymerase, option C, DNA ligase, option D, DNA lyase. So let us see, the question says, which of the following enzyme is molecular scissor? So we have to understand this terminology, that is molecular scissor. So what are called as molecular scissors? Molecular scissors. So have you heard about it? Yes, all of you would have heard because we have already done the synopsis of the chapter. So molecular scissors is these are nothing but the enzymes which have cutting ability, okay, which can cut the DNA. Okay, so usually we call molecular scissors which can cut the DNA at specific region. Okay, so these are the enzymes which can cut DNA at specific region. Specific region. And you have various types of molecular scissors which are also called as restriction enzymes. What are these called as? Restriction enzymes. And why are they called as restriction enzymes? Because they are restricted to their specific region, specific site on the DNA. So what do? Suppose you have the DNA like this, and you have the various nucleotides here, or the base pairs. So they have the ability to recognize a particular sequence and cut the DNA. That is why we call it as restriction enzyme, because they will cut at the restriction sites. These are called as restriction sites. Okay, restriction sites. So they are specific to the restriction site. Each enzyme is specific to its restriction site. And here, for example, if I have to give you, that is BAMH1. BAMH1 is a type of restriction enzyme which cuts the DNA at a restriction site, like at a specific site. So let us see. Option A says BAMH1. Yes, that is correct because this is a type of restriction enzyme. Restriction enzyme, which is also known as molecular scissors. So all the restriction enzymes are called as molecular scissors. So BAMH1, yes, it is a type of restriction enzyme. And then you have three types of, majorly you can divide restriction enzymes into three types: type 1, type 2, and type 3. And BAMH1 is type 2, you can say type 2 restriction enzyme. And this can, identify about, and it can cleave about 6 base pairs of stretch of the DNA. Okay, so that is BAMH1. So that is the correct answer. Option B is RNA polymerase. RNA polymerase ruled out because polymerase will not cut. Instead, they are involved in extension of the strand. And here, the extension of the strand happens of the mRNA and not DNA. Okay, hence it is ruled out. Option B is ligases. Ligases have the ability to join the nicks of the strands. Okay, suppose you have the DNA strand which is cut. So this cut strand will be nicked by, or sorry, it will be ligated by these ligase enzyme. So ligation is nothing but joining the two strands, the cut ends of the two strands. So here we are talking about cutting, not joining, hence it is ruled out. Option D says lyases. Lyase is also a type of enzyme which have the ability to break the bond. Okay, only the specific bond. Hence it is ruled out, because here we are talking about the entire, double-stranded DNA will be cut by the restriction enzyme. But DNA lyases, they can bring about chemical change in the particular site, okay, of a particular bond. Okay, they can break a particular bond, not the entire double strand. Hence it is ruled out. So the correct answer is BAMH1. So let us conclude the answer. So what is the answer? The answer is option A. I hope you understood the explanation. Thank you.